All right, guys, we're going to go ahead and get started here. I've actually got, so antibiotics are one of my favorite topics, aside from, that they basically keep most of humanity alive for long periods of times, but also because they're really interesting and cool. I've got a really cool science thing of the week this week that I'm really excited about, so I'll share that with you guys at the end. So we're going to talk about different ways to control bacterial growth. One of them is to use antibiotics. There's lots of different types of antibiotics. Unfortunately, most of them we've just stolen from other organisms. We haven't really come up with our own, but we've modified, we've made improvements. But our own like de novo antibiotics are not really a thing that exists. So right here you can see an example. This is like a tetracycline here, and it's got a binding region within these guys right here. So this is an actual like working, how does this actually work kind of situation. But we'll talk more about that in a second. Real quick, I don't know if you guys have seen, because sometimes the professors teach 4302, do show this video. Have you guys seen this video before? Yes? Okay. So I really like this video. If you haven't, go watch it on your own. But basically, it's a perfect example of how quickly antibiotic resistance can occur. One thing I do want to point out, which is actually really cool that they don't talk about in the video, but if you go and listen to interviews done by the guy who did this, there's two things that you guys don't realize that they talk about. The size of this plate, which is about the size of this table, it's huge, but they also put squid ink they used squid ink and India ink to make the black surface area because those are not something that are toxic to bacteria. So to create the black background, you have to have something that works really, really well. And of course, they did have one slight mess up with some spores landing on it, and it created a little mold pile in the middle of it. It was really interesting. But basically, the manufacturing of this and the demonstration of this is really cool. So, but we're going to skip that because you guys have all seen it. So you guys don't need to listen to that again for 2 minutes. So, all right, so a couple of things. Whenever we talk about types of chemotherapeutic agents, or basically anything that we give to humans, we need to know what the mode of action is. How does it affect the bacteria? What is it, or whatever it is, if it's a virus or bacteria or fungi, how is it working? What is it doing to them to cause them problems? Is it inhibiting or interfering with their metabolism? Is it killing something? Is it destabilizing something? What have you? You should also know the difference between bactericidal and bacteriostatic. Bactericidal means that it kills it. Bacteriostatic means that it inhibits its growth. So you might be thinking, well, why wouldn't we always go for something bactericidal? Well, bactericidal's not as common, and it comes with some other consequences potentially to us. Bacteriostatic, a lot of the bacteriostatic antibiotics that we have, which all they do is inhibit or slow down replication, are actually really good for us because we can take huge amounts of them and not have any bad side effects for humans, but it is a lot of bad side effects for bacteria, and then we let our immune system come in and take care of the rest of it. So that's kind of the trade-off for those, generally speaking. You also need to know what therapeutic index is. So a toxic dose is a dose that must be taken before you get undesired side effects in a patient. Therapeutic dose is the dose that must be taken before the desired effect is seen. Pretty simple. Therapeutic index is basically toxic dose divided by therapeutic dose. That is it. It is not a complicated equation. Now, generally speaking, the safer a drug is, the larger the therapeutic index it has. So if it has a therapeutic index of 100, like ampicillin, that means that you would have to take effectively 100 times more than whatever the therapeutic dose is to get a toxic dose. That's a pretty big thing. Something like gentamicin, though, has a therapeutic index of 3. That means that at 3 times a normal dose of gentamicin, you're going to start seeing severe side effects. And these side effects for gentamicin, for example, are pretty severe. So ampicillin, very, very safe drug. You would have to be taking what is effectively kilograms of it, which is really hard to do, to get bad side effects. So very, very safe, generally speaking. Gentamicin, not so safe. And some of it can just build up in your system, which is the downside. So therapeutic index isn't perfect because side effects might start popping up if you're on it for a long period of time. One of the ones that is a great example of this, because its build up is an important factor, when we had COVID first coming around, we had a lot of people who were taking a bunch of different things. One of them was hydroxychloroquine, which is used to treat a variety of other diseases. And one of the things that was most commonly misreported in the news was like, hey, it's an incredibly toxic drug, it can cause blindness. I'm like, well, yes, but no. So it does cause degeneration and issues with vision in people who have to take it for long periods of time. People who get treated for lupus and some other diseases will sometimes take it, and they'll be taking it for their entire life. And so you only start seeing people with those issues with their eyes at the 5 or 10 year mark. So about 5 to 7% of patients at the 5 year mark, and about 10 to 13% of patients at the 10 year mark will start developing symptoms of vision loss due to the drug, because you have to build up over a kilogram of cumulative medicine to get those negative side effects. So, well, yes, it can cause blindness. It doesn't if you're taking it for a normal, short course of time. So you got to keep these things in mind, the length of exposure, the total buildup, and then the difference between toxic dose and therapeutic dose. So if you're taking a huge amount of the toxic dose, then you'd get the side effects really quickly. But if you take it for a really long time, even if it's at a therapeutic dose, they can build up sometimes depending on how the drug works. Okay, so that's my little rant. So ideally, we always want to treat patients exhibiting the highest therapeutic index to minimize harmful side effects. Sometimes we have to choose low therapeutic index ones. A great example of this are things like fungi, because fungi are eukaryotes. And eukaryotes are like us. And so a lot of things that will target eukaryotic pathways will target ours. And unfortunately, a lot of things that work on eukaryotes really, really nicely tend to get stuck in our kidneys and livers, which is where we see a lot of damage from taking those drugs. There you go. Obviously, a lot of chemotherapy treatments tend to target cancer cells, which are basically our own cells, and so they share a lot of similarities. So a lot of times you get overlap and unintended cell death. So sometimes you have drugs where the therapeutic index is incredibly small because you're going to always get side effects, and sometimes you get side effects even if you take below the therapeutic index for the drug, such as in cancer treatments. So the toxic dose and the therapeutic dose are the same, or even greater. Okay. So that little rant being done, there's a bunch of different types of antibiotics. This class list, so it's a broad class list, isn't necessarily encapsulating everything. And a great example of this, I'll show you guys in a second, but basically we can fit them all into these big categories, which are sulfur drugs, penicillins, tetracyclines, fluoroquinolones, macrolides, aminoglycosides, and there's some wiggle room in there, and we'll talk about in a second. Well, actually, I'll just talk about it right now. Penicillins. So penicillins, penicillin, amoxicillin, ampicillin, these are all types of penicillins. But there's another type of drug that also fits in this category that doesn't quite look like penicillins. It's kind of like penicillins, but there's a whole bunch of varieties that they're not called penicillins. Does anyone know what that one is that looks like that? What is it? Nobody? May I have a guess? Really common antibiotic to prescribe, found in Neosporin. So it's the cephalosporins. Cephalosporins are antibiotic that are penicillin-based. They do and target the same thing, but they're kind of their own little class. They're weird. So there you go. There you go. So note that this isn't a definitive list of classes, but it's kind of the broad classes that cover the major topics. Okay. First one up, sulfas, sulfonamides. The one that's really, really common is sulfasoxazole, which obviously rolls off the tongue super easily. But what does it do? It's bacteriostatic by competitive inhibition of folic acid synthesis. Basically what it's doing is it's kind of getting in the way with PABA during folic acid synthesis for bacteria. We don't have to worry about that because we can consume it. We don't worry about that interfering with us because that enzyme process does not exist in humans. So we don't care if we mess that up because it doesn't matter for us. We do not care. So it's really, really good, has low side effects. The major side effects, well, okay, and by the way, folic acid, what it's doing is it's producing amino acids and purine, so it's considered bacteriostatic. You can still make those, you can still eat those from the environment to use them in your own proteins and enzymes and stuff. So it's slowing everything down. You just can't make it from scratch. The side effects, most common side effect is allergic reaction. I'm allergic to sulfa drugs. Doctors always hate it when I come in because I've got like a little cut on my arm or something. They're like, oh, we'll just give you sulfa. I'm like, yeah, I'm allergic. And I die, which is bad. So generally speaking, that's an example. Other ones are things like blood dichriasis, crystalluria, crystals in urine, hematuria, and thyroid hyperplasia. But more or less, these are incredibly unlikely ones to get. Crystalluria does happen, and I don't think we actually know the mechanism of why it happens as much. If I remember correctly, the thought was that sulfurs tend to like to build up urea in urine and form crystals really much easier. And so if you're taking a lot of sulfa drugs, you're just dumping a lot of sulfur into your system. But I don't think that we know for sure. Anyways, but basically crystalluria, and all that means is that it just is kind of painful to pee, and that's about it. But the allergic reaction is kind of serious, but more or less, you can take a huge amount of it, and it doesn't hurt you. It's considered broad spectrum. That means it handles both gram-positives and gram-negatives. So it's really effective. It's kind of the first hey, we can just give you this, you'll probably be okay. Which is why it makes doctors angry when they have to prescribe me something else. I don't really mind. It doesn't bother me at all. I understand why they rather give me sulfa. So penicillins are the next class. These include things like ampicillin, amoxicillin, penicillin G, penicillin V. How does it work? It's basically got this central beta-lactam ring, which is this guy right here. This is a constrained ring. In other words, if you guys remember your chemistry, nature's like, hey, I'd like to not be in this shape because squares are for losers and I would like to not be a square. And so it's pulling apart because you're basically trying to bend this into a nice little circle, and nature does not like tiny circles like this. It's like, let's not. And so it is ready to react with things to basically undo this. And the thing that it's ready to react with, most likely, is the cross-linking of NAM units in peptidoglycan molecules. So this works really, really well on gram-positives because gram-positives have peptidoglycan all over on the outside to hold their cell wall structure together. So if you start messing with their cell wall structure, then they just kind of explode. So this is considered a bactericidal antibiotic. So it is most effective against gram-positives. It does work on some gram-negatives, but not as well. There's a couple of side effects. These are basically all just allergic reactions. So if you're allergic to penicillin, that's basically it. The amounts you'd have to start taking to get other side effects is ungodly, more than a human could consume in a day conceivably. So most of these side effects are just, hey, are you allergic to this? If you are, you can get things like hives, you can get a rash, you can go to anaphylactic shock because it starts shutting down your airways, things like that. Fortunately, not a lot of people are allergic to penicillins, and even if they were, we have lots of other drugs they can take, which is good. So takeaway from this one, central beta-lactam ring, constrained ring, doesn't like to be like that, wants to break apart, and reacts really easily with gram-positive cell wall structures. The next one is tetracyclines. These ones are really, really cool. There's a multi-ring structure. Tetra for cycline, cyclical tetracyclines. Easy to identify. Okay. In case you guys ever have to memorize these by their shape and structure, unless you're a biochemist, I probably don't think you're going to have to, but in case you were wondering really hard how to do that, that's how you do it, tetracycline. Okay. These include different ones. The ones that you guys probably might be really familiar with if you work in a hospital setting or planning to is doxycycline, which is kind of the really commonly prescribed one. Minocycline, of course, tetracycline and oxytetracycline are the really common ones. I think doxycycline is probably the most commonly prescribed one. How does it work? It works by inhibiting protein synthesis by binding the 30S ribosomal subunit. This part's really important. Keep that in mind. So where's it binding? It's binding to the 30S ribosome. It's saying, hey, I'm going to come in here and I'm going to prevent you from bonding between the tRNA and the mRNA. So basically, every time you bring in a tRNA to bring a new amino acid in and make an elongated peptide chain, it has to come in and it has to dock with the mRNA to make sure that's the right amino acid. Well, this guy just kind of gets in here and he just kind of sits in that middle part and makes it hard to make that connection. So when you do that, you're basically slowing everything down. So it is considered bacteriostatic. It just makes everything go slower, which gives your body time to respond. It is considered broad spectrum, which is really handy for us. Side effects. Common ones are things like nausea, diarrhea. Diarrhea is a really common side effect of a lot of these antibiotics because it starts killing off the bacteria in your gut, and your gut needs bacteria to do things properly. The weird ones, discoloration of growth plates and unerupted teeth in children. So before we knew this, we would give it to children, very young age, and then they'd have their teeth come out and there would be weird banding that would happen, and that was because of tetracycline. We don't tend to give to children as much. Also, it can cause some other issues, but the big one, the serious ones, are damage to the liver and kidneys. You'd have to take a lot of this, and some people are more responsive to this side effect than others, but generally we'd be pretty kind of keeping an eye on that if you were taking it for long periods of time. The other thing about this that's kind of weird and wacky that you guys should note is it chelates in milk or calcium, so you should never take it with milk. So if you take it with milk, what it's going to do is it's going to come out of solution. Your body won't absorb it. It'll be functionally useless, so it's kind of a weird one. Okay. Fluoroquinolones. So these guys are really cool. You guys want to know how to identify them really quickly? This is a bunch of different types of fluoroquinolones, but they all have this kind of basic structure, which is they kind of have this two-ring structure. They have a fluorine in this position right here, fluoroquinos, and so they've got a nitrogen right here, and then they're going to have some other side chain sticking off on the left-hand side, and then sometimes something else coming off from the bottom here, but not always. And what does this do? Well, it looks a lot like adenosine, which I know if you're looking at this right now, you might be like, that doesn't look like adenosine. Well, it kind of does. It's got a little part that sticks out here, it's got a nitrogen here, and it's got a little ring right here, and then a big ring right here. So it kind of looks like adenosine. If you squint really hard and don't think about it too much, it does. So why is that important? Well, it inhibits topoisomerase 2 and 4, so it prevents DNA replication and transcription. So topoisomerases kind of unwind things and pull them apart, and they're really, really handy and cool things. So examples, ciprofloxacin and levaquin are the kind of common ones. Ciprofloxacin is probably the more common one, and enrofloxacin also. Big issues with this one. So it can cause erosion of cartilage in weight-bearing joints in children. Giving children arthritis at an early age because they took antibiotics, bad thing, so you have to be careful of that. The other one is tendon rupture. Now this one's actually really interesting, and I'm going to put this as a PSA for all of you upcoming doctors and physician's assistants and whatever else you're going to be doing in the medical field. Athletes, especially athletes in high contact or high impact sports, should not be taking this antibiotic unless they absolutely have to, and if they do, they should be reducing their training until they're fully off the antibiotic, because they can do a little juke move and have their tendon in their knee fall off because they've been taking this antibiotic, which messes with the tendon stuff. So it specifically messes with where the tendon interacts with the muscles. And there is actually a number of athletes that have gotten serious, serious injuries, and it's actually because they were taking an antibiotic that they didn't realize or think about having that issue. So just a little PSA for you guys. Okay, that aside. Other things can cause vomiting, diarrhea, things like that. Rash if you're allergic to it. It can also cause abnormal liver tests. So this is something that you don't want to give to somebody with liver failure or potential liver failure because it can mess with the way their tests are. So if you're monitoring them for that, it really kind of screws with things a little bit, so don't be careful with that. And of course, nausea, diarrhea, which we talked about before. Broad spectrum. So it is considered broad spectrum. It does work on a variety of organisms. Everybody has topoisomerases because they need to unwind DNA. It is considered bactericidal because if you cannot pull your DNA apart to replicate, you can't really make stuff, and you can't replicate because your DNA is stuck together. So kind of an important thing. Okay. All right, I'm going to go really fast. Anybody have any questions on the antibiotic so far? Makes sense? Yes? Okay. Macrolides. These are my favorite just because they're huge. They are large ring structures. They are really, really cool. The ones that everybody's kind of familiar with are Z-Pax or erythromycin. Those are the two commonly prescribed ones. There's a bunch of other ones. But erythromycin and azithromycin are kind of the really, really common ones. How do they work? Well, they're really, really neat because they bind to the 50S ribosomal subunit. So if you guys remember, we were talking about the other ones that bind to the 30S. These guys bind to the 50S. But they do it a little bit differently in just that they interfere with the elongation. They're just bacteriostatic. They're not necessarily like killing anything off. They're just kind of slowing things down. They work really well against gram-positives and some gram-negatives. So they're not considered broad spectrum because not all gram-negatives are susceptible to them, but they work pretty well. Part of this has to do with their size, and the larger they are in surface area, the harder they are to get through membranes sometimes. And gram-positive only has one membrane to get through. Gram-negative has two, so sometimes it doesn't lend itself well to going through it. Most common side effect, gastrointestinal upset. So diarrhea, nausea, maybe vomiting, probably not, but and then hepatic injury, which seems like a rather large jump to be like, yeah, we could destroy your liver. That's probably not going to destroy your liver, but it would potentially injure your liver if you're taking a lot of it for a long period of time. Less common. Usually doesn't cause liver failure or anything, just causes damage. So your liver enzymes might go wacky for a bit and your doctor would be like, hey, you shouldn't take it. We'll do something else. Okay, now, this guy, the more serious ones, aminoglycosides. Aminoglycosides are really, really interesting. This includes our guy, gentamicin, which we talked about before, which is kind of dangerous, has that really low therapeutic index. So is streptomycin, which also has a low therapeutic index. Streptomycin is actually really interesting. If you guys ever want to hear a really interesting story about how we discover antibiotics, the tale behind streptomycin and like the controversy among the people who are researching it is actually really, really interesting. But basically, we came up with a crazy idea to mass look through a bunch of bacteria in different soil samples to find ones that produced antibiotics, and we happened to find one, and it was really, really effective. What does it do? So its mode of action is to bind to the 30S ribosomal subunit and inhibit protein synthesis. But wait, I hear you say, we just had one that did that. Why do we have two? Why is this one different? Well, this one's different because it does something really crazy and wacky. It goes, hey, I'm going to bind to that 30S ribosomal subunit, but I'm going to go a step beyond, and I'm going to cause codon misreads. So codon misread basically says, hey, I'm going to let the wrong tRNA come in, bind, and put in the wrong amino acid into your protein. Well, if you put in the wrong amino acid into a protein, especially in the middle of a protein, that protein just stops working. Like 9 times out of 10, it just doesn't work anymore. It's really kind of it messes with the cell. And if it doesn't start working right away, tends to be if you make a bunch of proteins that don't work, they kind of fall out of solution, clump up, and when they clump up, they tend to want to stick to something, and they tend to stick to the inside of a cell, and when you stick to the inside of a cell, you cause them to lyse, which is bad for the cell. For us, it's great in this case. They are very, 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 very effective against gram-negative bacteria. They are considered bactericidal. And so if you guys look right here, here's that 30S subunit, here's the 50S ribosomal subunit, here is our tRNA coming in, and then up here somewhere is the protein. And then over here is part of this purple guy right here, that's that 30S subunit. And then right here, this green bit is the mRNA. And you can see that this guy likes to bind in this area, and he just comes out right here, and he just starts putting in the wrong stuff as you're going along. That's really effective, really, really effective way to stop it. Now, why don't we just prescribe this all the time? Well, it's got some serious side effects. So, allergic reaction, you can have a pretty severe allergic reaction to them. So that's one. And those pretty severe allergic reactions, I'm talking about like your throat starts shutting down and you start not being able to breathe really quickly, even with one dose. So that's kind of a problem. Other things, obviously nausea, loss of hearing. So it damages the auditory nerve. So you can just lose your hearing completely from just taking this antibiotic. It also causes renal damage. So if you take enough of it for long enough, your kidneys can start shutting down. If you have weakened kidneys and you take too much of it, it can cause your kidneys to shut down. If you have diabetes, chances are that you might have problems with your kidneys. And if you have an infection, your doctor might go, oh, we need to treat this infection really aggressively. Here, take some aminoglycosides. And now that you have kidney failure. That's one of the consequences for these. These are things you have to think about. So antibiotics, very confusing, lots of little different things you can mess up on. So how do we test antibiotics? We do a couple of things, and what we're going to be doing is what's called a Kirby-Bauer test. The way we do it is we use this antibiotic testing media called Mueller-Hinton agar, and we're going to do a 4-way streak on it. So we've been talking about 3-way streaks. 4-way streaks is a little bit different. All we're trying to do is create nice, even growth across the entire plate. We don't have to press hard. We just want to make sure that we don't have any open areas that have like large gaps in them, because we want the bacteria to grow in a nice, even lawn. So just like we did for when we did the gram-positive tests. So this is the Kirby-Bauer test. The media here is Mueller-Hinton agar. It grows in a very standard way, and it helps kind of even out the way bacteria grow on it, so it's really good for testing antibiotic resistances. And then, once you do the streaking part, you will then use a little machine to clip off antibiotic discs in a circle, evenly spaced out. And we have a machine that does that for you guys, and you guys will get to use that. It's pretty fun. And each of these antibiotic discs are labeled, and they are labeled with what antibiotic it is. So E, erythromycin, CYP, ciprofloxacin, G, gentamicin, so on and so forth. Not all of them are conveniently named. Some of them are like, you know, oh, what's this one? TET, what does it stand for? Sulfonamide. Okay. So some of them don't always line up, so you just have to be careful. It also has a number, which is usually the micrograms embedded into that of that antibiotic, because you need to know how many micrograms you're doing. Some testing ones will use different values, so these ones have it embedded in there. What you'll do is you'll clip that on there, you will let it sit for a little bit, and then you guys will put it in the incubator and let it incubate. Come back and look at it. You're going to measure the zone of inhibition, the area where bacteria do not grow. So you can measure directly across in a nice smooth line like this, or you can measure the radius if it kind of gets a little weird in some places. So you just measure the smallest radius and then double that for the diameter. And that's your zone of inhibition. A couple of weird things though. Sometimes they get a little hard to read. If there's little colonies growing into here, that means that that was not a complete zone of inhibition, so that is considered resistant. If it's growing right up to the disc and you got little colonies there, that's resistant. So you measure from the closest colony possible as your radius or diameter. Ciprofloxacin, you can see right here, there's this one zone right here that you guys can kind of see. It's kind of hard to see in the light, but you can see that one big zone. But then there's kind of like another zone where you see some bacterial growth in here. That inner zone is the actual zone of inhibition, because the first area has to do with the way the antibiotics spread out, and they just kind of slow down growth at that concentration. They don't necessarily kill it, and then it gets to the point where it's starting to be lethal. So you have to use the zone where you've got complete inhibition. Everything's dead, no growth at all. So in case you guys are curious, there's a way to measure this, and each one's different for each antibiotic. And this is actually, I kid you not, this chart and this chart are the actual charts that they package in with these discs so that we can read them. And they are literally that size font. It is terrible to read. It's not fun. But anyways, we have a chart set aside for you guys that has these. Each of these has different numbers, and it's really important to take these into account. You can just look at something and go, oh, that's a pretty big zone, that's resistant. Well, a great example of this is penicillin. Penicillin spreads out really quickly. So a zone of inhibition for penicillin might be, or let's see, there's another good example right in here. For gemafloxacin, right here, something that's susceptible is greater than 23 millimeters, but something that's resistant is less than or equal to 19 millimeters, right? But if you look at something like, oh, I don't know, intrapenum, it's resistant at 15 millimeters. And it's susceptible at 19 millimeters. So it starts its susceptibility at 19, whereas this guy starts its resistance at 19. So the size is dependent on each antibiotic individually. And sometimes these antibiotics are organism specific. So if you notice, some of these have different numbers for different ones. For example, the one for gonorrhea here is different than the one for Streptococcus pneumoniae, is different from the one for Enterobacter K and Staphylococcus. So we have to take into account which one we're using or which organism we're testing it on. Okay, Complicated, I know. So for you guys specifically, 4102, because you guys have 2 unknown organisms, the way you guys are going to do this is you're going to use your gram-positive for this unless, as long as that gram-positive is catalase positive. If your catalase-positive organism, if your gram-positive organism is catalase negative, those don't grow very well in these plates. So what we're going to do instead is we're going to have you guys use your gram-negative. So if you have a catalase-positive, gram-positive organism, you'll use your organism on your Mueller-Hinton plate. If you have a catalase-positive organism that's catalase negative, you'll use your gram-negative organism on this plate. And we'll have copies for those of you who had issues growing yours so that you guys will have a bacterial sample available to use for this. You will streak a plate, back and forth, do that, drop the discs on, start incubating it. Now, on top of this, we will also do something called an E-test, which is this guy right here. E-test will give you guys the bacteria to use for these E-tests. What they are is they are antibiotics embedded into this kind of weird nitrocellulose material, typically, or plastic polyvinyl sheet. And they're embedded in there at different concentrations, different amounts, so that you can determine what the minimum amount is that you have to give to kill that bacteria. So you just kind of measure whichever zone, right, where that zone of inhibition starts. So right over here, you basically see that there's no bacteria growing right at about 0.006 for DLX for this particular organism. This one's erythromycin, and you can see it stops growing right about here. And so you can measure these zone of inhibitions and the concentration that'll work for them for these bacteria, and these are really, really handy. In case you're wondering why they're called an E-test, that stands for epistomometer. Why does it stand for that? I don't know, but that's what they used to call these. And so there you go. Now, really important thing, when you guys put these down on a plate, you want to avoid putting them on the edges or like skewed. You kind of want to get them right in the center so you get a nice, easy zone of inhibition to read. If they're too close to a wall or something, it becomes confusing and doesn't always give you the best result. So try to get it right in the center. You will only do one of these plates per group of 4. So these ones are kind of weird like that, but one group per plate of 4. And so you'll just lay it down there with the organism that we give you guys for your group of 4. Okay, does anybody have any question on the E-test? Makes sense. All right, we're going to talk about our cool science thing of the week. This one's really cool. The title of it's actually really short, which is Programmable Protein Delivery with Bacterial Contractile Injection System. That doesn't make a lot of sense, but it's really, really cool. Now, this is a very new article. And when I say very new, I mean it was technically officially published to, what's the day, the 31st? On the 29th. So it's super new. I saw it and I was like, this is really, really cool. So if you guys remember, at the very beginning of the semester, we talked about a bacteria that creates a nice little glow in soldiers' wounds. Does anybody remember what genus that was? Totally forgot it. That's okay. Does anybody have a guess? Okay, it's called Photorhabdus. Photorhabdus asymbiotica. Well, these guys are actually really, really interesting. And what they do to communicate with each other and potentially with other cell types is they make these little tiny protein delivery systems that bind to specific items and then jam an injection tip into the cell and deposit a payload. And you might be looking at this and go, Josh, this looks identical to a phage. And that is really close. It is kind of similar to a phage. It's actually much bigger than a phage and way more complicated, but it's actually a lot of its principles are very, very similar. In fact, the binding end of it, the tail bit that binds here, very, very similar. This structure that makes this little circular thing, pretty similar. It's really cool. So we found these and we were like, hey, you know what? These are typically used to communicate from bacteria to bacteria, so we can transport signaling molecules across distances, and sometimes bacterial cells don't like to take up signaling things, so you can kind of like force the signal into there. We already know that sometimes these can make signals to other things like fungi, or we could take it to set signals to insect cells. So they're like, hey, this might be a really cool tool for us to use. I wonder if we can do it in mammalian cells or human cells. So that's what these guys did. So they sequenced this whole thing out, and they figured out basically how all the parts worked. They assembled it, so basically it's got this big multi-level structure in the codon region, the exon, and it's got structural protein. It's got these other ones which are like, I don't really know what they do, and then it's got this guy, which is a toxin, and this guy, which is a toxin, and then we got some other stuff in the middle here, and we don't really know what it does, but we can kind of cut it out, so it's okay. And so that's basically what they did. So basically they're like, hey, there's a system, there's this thing that we have, we kind of know how it works, we actually don't know how a whole bunch of it works, let's play around with it. And so what they did is they were like, hey, let's see if we can make this in E. coli. So they did a whole bunch of it. They did a negative stain of these purified particles and then scanned it on an SEM, and you can see these nice little long tubules here. They look approximately right when they're coming out of E. coli, just like when they come out of Photorhabdus. And the reason why we use E. coli is because E. coli is way easier to manipulate and grow, and Photorhabdus is finicky, so we just don't. We just skip it. And then we go, okay, hey, let's lyse these, let's run them on a gel. Hey, yes, these are making the right size things that we need. Cool. What are we going to do? Well, do these things still work? Okay, let's see if we can get them to bind. And we know this binding region right here at the bottom tends to bind to these insect things. So we have this insect cell line. Let's see if we can attach a glowy bit here and get it to stick there. Oh, yeah, it sticks. Awesome. And they're like, what if we can put glowing enzymes inside cells? So then they chopped out that first one because that's actually a toxin. It tends to kill a lot of things. So they're like, hey, let's put in GFP, Cre, zinc finger, and yeah, lo and behold, they were able to get those proteins in those cell types quite regularly. One of the things that they figured out, though, is that you have to have a cellular component, this PVC15, which is this purple guy right here, to load it properly. And that was kind of one of the big hurdles, because they're like, yeah, so there's this one structural protein in here. We don't know what it does. Well, it turns out it's involved in loading it in. So they're like, okay, let's try this out on a bunch of different cell things. So they're like, let's see if we can kill regular old insect cells with this. So here's just regular PBS. These cells are glowing as normal. We expect them to glow. The wild type 1 means that these guys are putting their little toxin in. That means it kills everything. If we mutate the loading protein, so PVC13, which is ATPase that basically helps load the contents into this little space capsule thing. If we mutate that, it doesn't work. If we mutate PCV15, which is one of the things that helps do the packaging, that doesn't work, then the cells live. Oops. And then they did a whole bunch of things where they're like, hey, can we plug plasmids in here and put plasmids into these cell types and get them to glow? So they tried it with PBS, they tried it with the wild type, and it made a whole nice glowing thing, which is great. Then they tried it with the mutated one, it doesn't work, and it doesn't work. So they're basically handling their controls. Controls are important. This one had a slew of controls in it. This article was so complicated, it did everything. So we talked about it, we go, okay, so it can infect insect cells, great. It can kill insect cells, great. We can modify it to put plasmids into insect cells, awesome. What else can we do? Well, let's go ahead and package it up with a bunch of proteins and a special tip and see exactly how much can we mess with different components of this. So one of the big things that they wanted to mess with is they want to see, hey, what's the smallest way we can basically get it to load proteins into here? So there's a region called the NTD region, which is really, really crucial for getting it in there. We know that you have to have that region, and then some of the other regions, but we didn't know how much. So they basically did a truncation mutation where they're just chopping off bits of this until it got smaller and smaller. They actually got, oddly enough, with a little bit of the gray part here, so this toxin bit, you just have a little bit of toxin bit, you get a really good loading here. That's awesome. With this guy, you can actually just chop off all of the toxin gene right here and just have the loading bit, the NTD region, and it loads really, really well. Awesome. So No problem. We can load things now with a really small sequence, which is really good for us because we don't want to put in too much protein sequence. Then they're like, okay, well now we need to get it to bind to other things. And here's the problem. The binding ends, we don't have an analog for them because we really only know that Photorhabdus binds into insect cells. So unlike phages, which have big giant pools and are mutated all the time, and we know how those tails work, we can kind of make a cross-lap, but we don't have the foundation. So they were like, hey, let's just pretend that they work exactly like the T-phage cells, or the T-phage ends. We're going to use a computer-assisted modeling system to cut off ends and then figure out how much we can put on there. So they basically made a whole bunch of mutations. They basically were able to say, okay, we're going to cut off the entire bottom here and make these little linkers here. So these linkers normally would be extending into here to this bit. They just cut it off and put linkers in. When you cut it off and put the linkers in, so it's truncated, it doesn't bind anymore. It doesn't work. So that's bad for us. But the linkers now mean that we can attach things onto it. And in this case, they put in adenovirus 5 knob, which is a binding region for adenovirus to infect humans. Turns out you can infect human cells with this now. Wow, works great. They're control. Hey, let's mutate a small little part of the adenovirus 5 knob. Now it doesn't work. Awesome. We already knew that it shouldn't work if we make that a mutation, and now we can show it doesn't work for this system. Let's do it with another binding region, which is for DARP-in. DARP-in, oh, it works really, really great. Awesome. Let's do a little tiny mutation for this binding region. Oh, now it doesn't work. Okay, great. So now we can infect mammalian cell types, specifically human ones, but we can infect other ones too. And so they're like, great, this works really, really well. Let's do this. So they started doing this experiment and a whole bunch of other ones. And then one of the really wacky and crazy things they were trying to do is this guy right here, where they're like, let's put in a bunch of different types of packages into this. And one of them they made two mixtures, one with this thing called zinc finger left and one with this thing called zinc finger right. And then they put both of those in a big mixture with cells. And if you have zinc finger left and zinc finger right, you should be able to make a mutation because the zinc finger is part of this mutagen protein, and you get this huge spike in the gene being mutated. But if you only put in the one of them, you get nothing. So you can actually just make mixing and matching of different proteins, all in the same cell types, and they work in combination. Even better. Then they were like, hey, how far can we stretch this? Let's try it with a whole bunch of different types of binding things. And basically they did things of like, an HA tag, a flag tag, an EE tag, moon tag, sun tag, alpha tag. It works in a whole bunch of different situations. Basically, if you don't have the ability to infect that cell, if you're not specific to that target molecule, you don't work anymore. But if you do have that target molecule, you're really, really, really specific. So this was like a really, really complicated test, basically just to show, yeah, if you have the targeting bit for this guy, you will only be glowing with this guy if you have that targeting bit, and everything else doesn't work for that guy. Awesome. And same thing for over here. Oops, is that my last slide? I guess it was. It's the last one. So they did this with a bunch of human cells. They basically were able to put a whole bunch of proteins in the human cells. Really, really cool. The last one they did with live mouse. So the biggest hurdle with any kind of drug thing is, hey, can we test in living things? So they did it with mice. And what they did is they put in these mice that had already had genetic mutations for this thing called LOX T tomato, which makes a red protein. If you have a thing that clips the gene and flips it right side up in the mouse brain, it'll make this red protein. So they loaded up these little PVC container deals with a bunch of the enzyme, which is called CRE, into there, injected them into mice. It went up into the mouse brain, and they got a clipping, and they got the mouse brains to glow red with this. So in a living mouse model, they were able to actually make this work. They're able to put proteins exactly what they want and do exactly what they want and do a huge thing. Some of these things, especially zinc finger is a huge protein. The larger protein that you can put in, the better it is. This is actually like really, really handy because we can put proteins into humans. but it's really hard to get proteins to get inside of cells to do something to humans. With this system, we can put proteins inside of cells, and if they're inside of cells, that can let us do a lot of things that we couldn't do before. So this system's brand new, really, really cool, cutting edge, really cool. You guys probably see something about it later on in your careers, but I just want to tell you guys because it's so interesting, and I know it's really complicated. What you should take away from this: we stole this system from Photorhabdus. It packaged things in like what looks like a T4 phage, but it's a thing made purely out of protein by bacteria, and it jabs it into other cells and loads protein into the cell. And we can manipulate that to do proteins into other cells that we want, and we can make it specific to cells that we want to look at. There you go. All right, any questions on this one? I know it was kind of complicated, but that was really cool. If you guys have any questions, let me know. I'll see you guys later. Have a safe week. And we'll get started with antibiotics next week. Yep.